this is where we're going to talk about the cells. So a unifying concept in biology would be the cell theory. And cell theory states that all living things are composed of cells. Organism can be unicellular, can be multicellular. Cells are the basic unit of structure and organization of all living organisms, and they can only arise from pre-existing cells. There's no such thing like spontaneous generation. So it means cells have to come from pre-existing cells. And if you wonder how big cells typically are, notice these are eukaryotic cells, animal and plant cells, compared to bacteria. So notice bacteria basically are the size of mitochondria. And here we discussed our macromolecules. And just to give you an idea of how big viruses are, even though technically they're not considered living things, you can see they are smaller than bacteria. So why do cells remain small? Before they get any larger, they go on and divide, given that they receive the signal to initiate cell division. So as you already know, cells are the basic unit of life. So they have to be able to remain alive, and they need to be able to maintain homeostasis. They need to be able to obtain nutrients, eliminate waste. So there will be a lot of transport happening across that cell membrane. So when we look at the plasma membrane, cell membrane, we are identifying it as the surface area. So it has to be, that surface area, plasma membrane, has to be large enough for efficient transport to occur. We compare the surface area to the volume, which is represented by the inside of the cell. So it means we have all the molecules and all the organelles within the cell, lots of metabolic activities happening within the cell. So if the cell is really, really big, that means the demand for such transport is going to be huge. So what we need to be able to do here is calculate the surface area and volume. How to do that, the formulas are given to you on the exam sheet, so you do not have to memorize them. So the only thing you need to do is be able to carry out the calculation. So here's an example. We have a 1 4-centimeter cube, and if we do the calculations of surface area to volume ratio, we're going to see that this ratio is 1.5 to 1. And then we have cut that 4-centimeter cube, and now we have 8 2-centimeter cubes. So we can see one of these cubes will have 3 to 1 ratio. So it looks like the surface area is increasing as the cell size is decreasing. And then if we divide this 1 cube again, notice we're going to get 64 1-centimeter cubes. So now this little guy right here will be 6 to 1. So it means the smallest cell will have the greatest surface area to volume ratio. Therefore, the smallest cell in this case is going to be the most efficient when it comes to transporting materials across the membrane. So now remember, as cells increase in size, the surface area to volume ratio decreases, because when the cell is increasing in size, the volume increases much faster than the surface area. So once again, we want to keep those cells small to get that efficiency, what we're looking for in the transport. So cells are going to be adapted to function, and they will come in different shapes and forms to maximize that efficiency of the transport. And as you can see here, we have a root hair. So this is just one cell, but you can see how elongated it is. That's because you want to maximize the surface area, and this is where the water is coming in into the plant. So the more membrane you have, the more aquaporins you have, the more water will be able to come in. So that transport is going to be much more efficient and rapid. Another example of such adaptation would be small intestine epithelial cells, and they have microvilli. I don't know if you can see it here, but they actually draw it like this. They fold these little projections like so. So here it is, because the absorption of nutrients is happening here in the small intestine. So if you have more membrane, that means these molecules can pass through that membrane easier and much faster. So again, we're talking about the efficiency in movement. So now we have two basic cell types. So we have prokaryotes and eukaryotes. So we characterize prokaryotic cells by having no nucleus. Yes, they have DNA. DNA is sitting in the nucleoid region, but there is no nucleus. And also what they do not have is membrane-bound organelles. So when we say membrane bound, that means organelles that do not have their own membrane. So yes, they have ribosomes, so it means they're fully capable of producing their proteins. So now another thing, notice that bacteria have cell wall. And we talked about the fact that plants have cell walls and fungi have cell walls, except that their cell wall chemical composition is going to be different. And then here is for you to appreciate the diversity of bacteria. And notice they come in all different shapes. So we have round shapes, so we call that cocci. We have rod shape, bacilli, and some of the photosynthetic bacteria here that can actually produce oxygen for us. So here we're looking at two different types of eukaryotic cells, and we have animal cells and plant cells. So what I would recommend here, you pause this video and look at all these subcellular structures and organelles and see what structures are present in animal cells and what structures are present in plant cells, and see if you can see any differences. So there is what we call a endosymbiotic theory. So basically how membrane-bound organelles have actually evolved from previously free-living prokaryotic cells. So this theory is supported by the evidence, and some of the evidence we have is basically that these organelles right now, mitochondria and chloroplasts, they actually have a double membrane. So they have the inner membrane and they have the outer membrane, so two membranes. They also have free ribosomes, and they also have circular DNA molecules. Clearly, if you have DNA and you have ribosomes, that means these organelles are capable of their own protein synthesis. So they seem to be pretty independent. And also, in the way they grow and reproduce, they resemble prokaryotic cells. Basically, they grow to a certain size and then they split in half by binary fission. So I do have a story of endosymbiotic theory, so this is very self-explanatory. I would like for you to pause here and read this paragraph on your own. Alright, so now we're going to move on and talk about the actual cells and their little tiny compartments. So every single cell will have a membrane, and we're going to talk about the membrane structure and function in more detail in part 2 notes, but right now just understand that this is going to be this boundary between the cell and its external environment. And lots of traffic is going to be happening within that membrane because cells are trying to stay alive and they're trying to maintain that stable internal environment called homeostasis. So the first compartment that we're going to discuss is going to be the nucleus. So if you look in the nucleus in the eukaryotic cell, you're going to see that DNA is wrapped around proteins. Then there is a nucleolus, which is that dense region. This is where ribosome production is happening. And then, of course, once the ribosomes are synthesized, they're going to be exiting the nuclear pores because they need to go into the cytoplasm and also attach to the ER. And now the liquid inside the nucleus is going to be called nucleoplasm, just like cytoplasm in the cell overall. And then what we have here is a double membrane, and we call that nuclear envelope. So now a little bit about the organization of the DNA inside the nucleus. So when the cell is not dividing, the DNA is going to present itself in the form of chromatin. So basically what this is, you have a DNA which is 2 nanometers thick, that's going to wrap around histones. And you can see there are actually 8 of them that will come together into a nucleus like so, and then the DNA is going to wrap around them. It will make what we call beads on a string. They even make that 30 nanometer thick fiber that we call chromatin. So when, like I said, when the cell is not dividing, you're going to see these stringy, spaghetti-like looking fibers within the nucleus. So when the cell receives a signal to divide, chromatin is going to condense, meaning chromatin is going to start to coil and loop more and more and more and more, and finally we're going to be looking at what we call chromosomes. So they kind of look like X-shaped structures. You can see they are very, very tight and condensed. But the reason they are like that is because after all, during cell division, you want to separate that genetic material. So if you recall, spindle fibers are going to be attaching on one chromosome end and the other, and these two separate DNA molecules are going to be separated. So one goes in one cell, the other one goes in the other cell. So it means if we have a nice tight structure, we can separate that genetic material without making any errors. But if we had chromatin, where you have these fibers that are situated all over within the nucleus, you would have a lot of breakage and errors happen during cell division. So ribosomes are known as protein factories. So they are synthesized in the nucleolus, and they are not true organelles because they don't have their membrane. They don't have their own membrane. They are basically made up of protein and ribosomal RNA. So the ribosomal RNA is embedded in that protein. So this would be rRNA string, and then you also have another one right here within. And the ribosomes are going to carry out protein synthesis, and we're going to get to that in Unit 6. So now the next thing we need to discuss here is we have what we call endomembrane system. So as the word suggests, we have endo, that means inside the cell, and then membranous system. So all of these structures, subcellular components, are going to be making up this system, and they're going to carry out specific tasks. But the overall goal is basically to synthesize a protein and then modify the protein and package that protein and ship it to the final destination. So here is your big picture of how this process works. So this involves nuclear envelope. You can see continuous with endoplasmic reticulum. Then we talk about the Golgi body. We talk about the lysosomes, vacuoles, and plasma membrane. So we begin with endoplasmic reticulum. So as you can see, endoplasmic reticulum is continuous with nuclear envelope, and it forms these folded cavities. And this is where you have all the ribosomes attached. So ribosomes attached to the ER, and this is where the protein synthesis is going to be taking place. We actually have two ERs. We call that smooth ER and rough ER. So let's compare the two. Smooth ER lacks ribosomes, that's why we say it's smooth. It doesn't have that bumpy appearance. And it's typically involved in the synthesis of lipids and steroids. It can also metabolize carbohydrates and detoxify drugs and poisons. If, for example, we have liver, an organ that's responsible for body's detoxification, that means liver would be rich in smooth ER. Now, rough ER, on the other hand, we call it rough because you have the ribosomes that are attached to the surface of it. And as you can see here, ribosomes are synthesizing the protein. So protein is, or this polypeptide chain is being fed into the cavity of the ER. This is where the protein is folded, glycosylated, meaning sugar chains are added, and then a vesicle is going to form and enclose that protein. And then once the vesicle is pinched off, is going to travel to the next destination, which is usually going to be Golgi body. So you can see how ER can actually form a vesicle with the protein inside and ship that vesicle to the next destination. So just in case you wonder how these actual vesicles form, you can see there's a lot going on. Once the protein is produced in the lumen of the ER, they're going to attach to cargo receptors. And they're going to signal all the other proteins to come to the scene, lay down on the surface, and sort of form that pinching, as you can see in this picture. It's a very elaborate and very orchestrated event to form that vesicle. So Golgi apparatus, or Golgi body, is another membranous structure. It consists of flattened membranous sacs, and we call them cisterna. So the job of Golgi apparatus is to modify products of the ER, sort them, package them into vesicles, and then we're going to notice that we have three distinct regions of Golgi. So the cis face is facing the ER because that's where the vesicles are coming from once they are pinched off. And then they're going to move along through the medial cisterna, which is that medial region, and then be modified. Then they will end up in trans face. And so this is that region where the pinching of vesicles occur again. And these vesicles are going to the plasma membrane or they can actually become lysosomes. So it just depends on what type of protein was produced and what the purpose of that protein was. All right, so now we're looking at lysosomes, and remember, lysosomes, we get them from when the Golgi is pinching off vesicles. So lysosomes will contain many different types of hydrolases, and these are the enzymes that are used to break down macromolecules, and you can see some of the examples here. Nucleases, for example, will break down nucleic acids, proteases, proteins, glycosidases, glycosidic linkages, and sugars, and so on. And the pH is typically lower inside the lysosomes, and this low pH is maintained by this hydrogen pump. So this is an active transport where they hydrolyze ATP and they can move hydrogen into the inside of the lysosome to maintain that low pH level. You can see cytosol typically will have this neutral pH level here. So some of the functions of lysosomes, remember, we want to digest material that's been taken in by the cell, as you can see here. For example, we have a macrophage, like a white blood cell, that's engulfing a pathogen, and then that pathogen needs to be broken down and neutralized. So it forms as what we call a food vacuole. The membrane folds, surrounds a pathogen, and brings that pathogen inside, and the lysosome is going to fuse with that food vacuole, and digestion is going to occur. And then here's another example of how lysosomes can actually recycle intracellular materials. So it means we have a peroxisome here in this case, and also mitochondrion that's no longer functioning. So now lysosome is fusing with the membrane that holds those two worn out organelles, and digestion is happening right here. So vacuoles are basically your storage compartments, and plant cell, fungal cell may have one or several vacuoles, and yes, they are derived from endoplasmic reticulum and Golgi apparatus. So now, contractile vacuoles are often found in protists, unicellular organisms, such as, here's an example of paramecium. So it lives in pond water. Pond water moves in through this membrane, and then paramecium actually collects all that water, and then its contractile vacuole will be able to expel the excess water. So it's able to maintain that balance of water. Otherwise, if it didn't have this type of adaptation, paramecium's membrane would just rupture. So central vacuole is very big, and it's found in plants, and it takes up a lot of space, and it gives rise to that turgor pressure in plants, meaning when a vacuole fills up, it exerts a pressure against the cell wall, so the plants can actually stand up nice and firm and rigid, and they look beautiful. Now remember, animal cells, if there's too much water, they would actually burst. So now, when it comes to energy transformation, we have two main organelles that are going to help transform the energy. So in mitochondria, we are transforming chemical energy. So basically, we'll go from chemical to chemical energy. So what chemical energy are we talking about here? If you guys remember, cellular respiration involves glucose and oxygen, and then those two components, many steps happen in between, are going to be converted into CO2 and water. And behind the scenes, during that oxidation process, mitochondria generates lots of ATP. So that's the process, or the conversion of energy, that takes place within the mitochondria. So as far as the structure of mitochondria, notice it has two membranes. So here's the outer membrane, and then you can see the inner membrane is highly convoluted, highly folded. Why? Because it increases the surface area, because within that membrane, within that inner membrane, what we have here is electron transport chain proteins that are going to be involved in the production of ATP. So it means the more membrane, the more proteins, and the more proteins that are doing the work, the more ATP you can actually make. So as we talked about before, endosymbiotic theory, remember these guys will have their own DNA and ribosomes, because they used to be free living organisms. Chloroplast also does energy transformation, so it means it actually converts radiant energy into chemical energy. So radiant energy is coming from the sun, and then what happens here is the synthesis of sugars. So that's chemical energy that can be further broken down, energy extracted from that. So they're gonna be making their organic compounds that way. If you look at the structure of chloroplasts, you will also notice two membranes. So there's an outer membrane and there's the inner membrane. And then within that inner membrane on the inside, you're going to see stacks and stacks and stacks of folded membranes in the form of these little sacs. And these are called thylakoids. So the membrane of the thylakoid contains pigments. And these pigments capture light. So when the light is captured, energy is captured, electrons are excited, and the whole process initiates a photosynthetic reaction. So that's why we say chloroplasts are able to synthesize their sugar molecules. And then the liquid that bathes all these thylakoids is basically stroma, and this is where the sugar production is going to be taking place. Okay, so we're going to have a discussion in more detail about mitochondria and chloroplasts when we get to metabolism units. So this is just a quick introduction to the two organelles. Peroxisomes are known for oxidation, and they have a single membrane, not a double membrane. And typically what they do, they remove hydrogen from toxic compounds. They add the hydrogen to oxygen, turn that into hydrogen peroxide, which is, by the way, also toxic to the cells. However, immediately they can convert that hydrogen peroxide into water and oxygen. And of course, those two molecules are harmless. So, of course, if you're going to look at liver and kidneys, you're going to find lots of peroxisomes there, because that is the job of these two organs. They are also involved in photorespiration, which is a competing reaction with photosynthesis. Okay, so now if we take a look at the cytoskeleton, this provides support and motility for the cell. So basically, we have these highways, protein fibers that extend throughout the cell, and they carry out functions such as maintaining the cell shape and allowing the cells to move. And oftentimes it interacts with motor proteins, as you can see right here. This motor protein is literally walking down this highway, which is that cytoskeleton microtubule, and it's pulling that vesicle. So yes, when these vesicles are pinched off, when those motor proteins can actually pull them along. You know, as I said in class, when you look at yourself in the mirror and then you say, you know what, I don't like this about myself, I don't like this about myself. You know, you have these moments sometimes, right? No, just stop it right there. Just stop it. You have to really, really appreciate yourself on a cellular level. Your cells are working so hard to keep you healthy and beautiful. And when you get sick, they immediately go to work to restore that homeostasis and that balance within your system. You don't even have to ask them. So please do not insult these little walkers that are working so hard to deliver the cargo where they need to go. All right, so now we have three types of cytoskeleton fibers that give support and structure to the cell. And one of those fibers, we call that microfilaments, and you can see they're made up of actin subunits. They kind of intertwine in the form of a rope, and microtubules would be another type of fiber. Notice these guys are made up of tubulin dimers. So there's alpha and beta. They kind of come together in these protofilaments, and they form this hollow shape, hollow tube, making up this fiber. Intermediate filaments, on the other hand, they look like a spider web throughout the entire cell, and then all the organelles can kind of sit in place where they supposed to. So dynamic instability of microtubules refers to the fact that microtubules can be polymerized and they can be depolymerized. So whenever the cell needs to change shape, it's going to involve polymerization and depolymerization of microtubules. So what does this mean? When we want to elongate that fiber because we want to move forward, we want to form that extension. If the cell is trying to move forward and it's just one cell, you know, it has to change shape. So it means we want to extend that microtubule. So it means we're going to be adding tubulin dimers and build it up. It's called a rescue stage. And then when we want to shorten, if you want to shorten the other end of that microtubule because you need to sort of move forward, we're going to do depolymerization. And you can see how tubulin dimers are literally falling apart. So we call that catastrophe. So it means tubulin dimers can actually be recycled. Centrosomes and centrioles, So centrosome is basically the region where we have centrioles located, and centrioles will help with cell division in animal cells. Plant cells don't even have them. Cilia and flagella, of course, that's for locomotion and movement. So cell walls are present in prokaryotes, fungi, some protists, and plants. The chemical composition of the cell wall is going to be different among all of them. For example, fungi will contain chitin. Plants, protists will have cellulose, and prokaryotes may contain what's called peptidoglycan. Animal cells do not have cell walls, so they have what's called extracellular matrix. So it means all these fibers are going to be coming together and forming this matrix to provide cells with structural support and also sort of connect them into tissues. As you can see here, we have a membrane, and then outside that membrane, extracellular space, notice we have all these fibers that are made up of glycoproteins called collagen, proteoglycans, fibronectins. So they're all there to sort of bind these cells into tissues, and then of course you have tissues coming together into organs. So neighboring cells and tissues and organs, organ systems, they need to be able to talk to one another. They need to be able to make close connections, adhere, interact. So through that physical contact. So therefore, we observe these different types of cell junctions. So for example, in plants, this type of junction would be plasmodesmata, which is basically a channel that allows for cytoplasm to flow through from one cell to another. So it means they can actually share some of the cytoplasmic content. If they have some nutrients in the cell, they can send these nutrients to another cell. So therefore, we say that plant cells sort of work as a community, more somewhat together like so. Animal cells also have different types of junctions, so I have them listed here. The first one, tight junctions. So this is where we're going to observe these watertight connections, which are basically diffusion barriers. Usually epithelial cells will have these type of connections, these watertight connections. Another example, or any type of tissue that should have a really nice solid barrier that would prevent the cytoplasmic leakage in any kind of way. Desmosomes would be another type, and these are going to anchor the cells into sheets, and typically they are found in tissues that are subject to mechanical forces, like for example muscles, blood vessels, even the sacs of lungs. And you can see proteins here are going to sort of adhere. Let me erase that so you can see how they connect these two cells together into these strong sheets. And again, we call that sort of like a button-like point. So another example would be gap junctions. So this would be similar to plasmodesmata that we observe in plant cells. And these channels can actually be opened and closed. They are highly regulated, and oftentimes we see these type of channels of pores that allow to move ions and sugars and amino acids through. And of course, it's going to be super important in cell communication. And then we see these types of channels in heart muscle. So when cells need to be depolarized, you have to move ions so that all these cells together in a tissue, in a heart, can start beating. Okay, so this will complete our discussion about the organelles and their functions, and the next part is going to be about cell membrane, structure, function, and also transport. I'll see you then.